This is a production of Cornell University.
Okay. So today I'm going to talk about different things. But before I start, thank you, Jen, for the introduction, and thank you all for coming for this talk. Now, okay. As this picture indicates, I'm going to cover a lot of ground today. I'm going to talk about not a part, but most of my research program. And I'm going to try and attempt to do that in the next 45 minutes. Should start my timer. Okay. So there will be a lot of projects being talked about, and I'm going to give you a bird's eye view of the different projects. But if you want to know more in detail, I'd be happy to talk about that later today or anytime. Before I get into the research part, I'd like to thank or acknowledge a lot of people who have helped us with the work. So starting with people in my lab, postdocs, lab techs, uh, grad students, undergrads, RU students, all of them have done all of what I'm going to show is actually done by them. So all credit goes to them. We have had the fortune of having a number of collaborators in plant biology. I don't know, I should use maybe this. Lot of collaborators in plant biology, in SIPS, in Cornell and outside of SIPS, as well as outside of Cornell. So I appreciate and acknowledge their guidance and help in all our research. Special thanks to Frank Schroeder, he's down here, but Frank Schroeder at BTI, he let us use his mass spec for the last 7 years, and we've run thousands, if not low tens of thousands of samples on the mass spec. So every morning when I wake up, I pray to the longevity and safety of the mass spec. So as you see, a lot of data generated using that. And thanks to the funding agencies that have funded this, all of the research. So my lab works on plant metabolism. And personally, I like this topic because it is at the interface of both basic and applied research. Plants make a lot of natural products, which we use in our daily lives, but they don't make it for us to use, right? They make it for their own use. And because of the diverse roles of these metabolites, not only does it satisfy my intellectual curiosity, it also, for plants, it produces a huge number of, they produce a huge number of compounds. It's estimated that across the plant kingdom, there were 1 million metabolites produced, and individual plants produce thousands of metabolites in different organs. This diversity is because of the complexity of the underlying metabolic pathways, right? We have barely scratched the surface of both the metabolome as well as the genetic networks that produce this diversity. That's what my lab has been focused over the last 7 years. And because there are a number of projects, we have classified them into 3 major types. First of all, figuring out what is the scale of this diversity, how is this diversity produced at the evolutionary and biochemical level, and how can we use it for our own purposes. We work with a number of model systems, and I'm going to talk about some of them in this research. So the next few slides are organized in this manner. So if I'll talk about the 3 areas and then end with just one slide on summary and future plans, although each of the areas also contain what we are currently working on. Starting with metabolomics. Uh, Now, as I said, we do a lot of mass spec, and it's a lot of people do transcriptomics and genomics, but there are some challenges associated with metabolomics. The first is that the name itself is a misnomer. When we do metabolomics, we are doing basically metabolite profiling. When you do transcriptomics, you are getting most of the transcriptome expressed at that given time. But in metabolomics, because there are so many diverse chemical structures, we have this compound which has a lot of hydroxyl sugars, but these carotenoids do not have anything that will enable it to solubilize. It's a long hydrocarbon chain. And so the type of extraction solvent you use will determine what sort of compounds will be extracted, right? Then they have to move in an electromagnetic field to get ionized. And the type of mass spec you use, the parameters you set, the masses you set will define what sort of compounds you will detect. And then the type of column chemistry you use will also determine how well you are able to separate those compounds. So the first challenge here is when you are sampling the metabolome, you are getting only a small part of it. And even if you get a small part of it, when it comes to annotation, less than 5% of the peaks can be annotated. So in transcriptomics, or when you have a gene sequencing Sanger sequence, you can blast it again in CBI, you can get the domain, and you have some information about what that gene does, looking at it's a transcription factor or whatever. But what about this 95% of the metabolome for which you don't know anything? So that's what we tried to do. We have been trying to work on as part of our research, looking at if we know the fragmentation pattern of these metabolites, can we get better insights into this 95%? Okay. I'm going to give you this case study of resin glycosides, which are structures, chemicals produced in convolvulaceous species that include sweet potatoes and some other horticulturally important crops. This resin glycosides comprise of this oligosaccharide chain to which you have these acyl chains attached and this long C16 fatty acid with hydroxyl chain attached. And there are lots of variations upon this basic structure. Uh, these compounds are important against various insects and nematodes, and they also have medicinal properties. So um, what we figured out was there is some pattern in how these compounds fragment in a mass spec. The masses, these can change a little bit, but we can predict what sort of masses they might have. So if you computationally dig into the mass spec data, can we recover more compounds this way? That's what Lars and Ali did. In my lab, they were looking at 30 different species of the Convolvulaceae family. And you can ignore all of the structural details. We were able to extract a lot of features from the mass spec data. This first column is important. This shows how many resin glycosides were discovered in each species. Now, just to give you some context, since 1992, when these compounds were first discovered, there are about 300 resin glycosides known. And in one species itself, we can find over 330 resin glycosides using computational analysis, and we can get a lot of structural information of those. So using this analysis, we are able to make better sense of the metabolites that exist in the metabolome. Now, initially, we were doing this in a very stepwise manner. We used to click on each peak in the mass spec data and then look at it manually. Then we started writing some Python scripts to dig into the files. That was also a little faster, but still slow enough. Last year, Vishwa, who is now a first-year plant biology student in Claire Castile's lab, he developed this website where you can just now upload the mass spec data files, and within a couple of seconds, you're able to get all the resin glycosides identified. So this simplifies the workflow quite a bit in our analysis. Now, what sort of biological information can this diversity reveal? Here, this is called a MSMS molecular network of all the resin glycosides we could detect across all species. The green color indicates the resin glycosides found in convolvulus genus species, and the purple ones are those found in Ipomia. And you can clearly see the diversification that has occurred between these two genera. That speaks to some biochemical diversity. Um, because we have the structural information, we can see what sort of pattern exists. And in Ipomia species, we see low molecular weight resin glycosides. In convolvulus, we see high molecular weight resin glycosides. And we also see the acyl chains that are added, they also vary. So we are trying to dig into these aspects more. And this also enabled us actually to select some species for RNA-seq because we know the tissue-specific differential patterns. Now, Baswati, who is a grad student in the lab, is looking at different accessions of sweet potato because these are connected with nematode defense. And she's trying to figure out how the diversity of resin glycosides vary. And then Irfan, a postdoc in the lab, is looking at the biosynthesis of these resin glycosides, so P450s, BHDs, glycosyl transferases that are involved in resin glycoside biosynthesis. So you can engineer them in other plants that do not have these compounds. So what these results show is that if we know the MSMS fragmentation pattern, we can make a lot more sense out of the diversity, right? We have shown this for other metabolite classes too, anthocyanins and flavonoids in different colored sweet potatoes, and then acyl sugars in Solanum habrochaetes. And I'm going to talk about this story in a few slides. later. But basically, it can be done if we have MSMS fragmentation knowledge, we can apply it to get more compounds annotated. But what if you don't know what the fragmentation patterns are, or if the patterns are not consistent? We have tried to analyze or champion two types of methods. One, identification-free analysis, which is basically multivariate statistical methods. And then instead of specific compounds, we can look at compound classes. So for example, how are flavonoids changing under stress, or how are nucleosides changing, and so on. This work was done by Liz, who was a plant biology student, now a postdoc at MIT. She was working on Brachypodium in collaboration with Olena's lab and Maria Harrison's lab and Joint Genome Institute. So we grew Brachypodium under different conditions, environmental conditions. These are hydroponic conditions. These were grown in some special sand soil mixture from where we could isolate roots, and these were grown in regular soil. And the goal was to get RNA-seq and metabolomic data and establish some correlations to predict gene function, or annotate gene function better. Um, Now, we did some preliminary processing with this data, and I'm going to just show you a couple of, this whole paper is like 30 supplementary files with 9 figures, but I'm just going to show you two figures from there. One is this diversity and specialization, which are basically metrics derived from ecological theory, or actually information theory, which diversity implies how diverse a given metabolome is in terms of the it's like Shannon diversity, how many peaks are there, how many, what's the relative abundance of different peaks. And we see leaves are much more diverse than roots metabolically. But we also saw that roots are more responsive to different stresses. We see that the specialization, this is roots grown in a specific hydroponic condition. And when we apply heat stress, the metabolome becomes much more specialized. There are many more unique metabolites that show up under heat stress in the roots, which does not occur that much in the leaves. This is another representation of the same. Here I'm showing a metric called RDPI, a relative distance plasticity index. And it basically measures how much the entire metabolome is changing, up and down together combined. How much is it changing in response to a given condition compared to the control? So the first 5 columns are the leaves, and the next 5 columns are the roots. And the same thing for the transcriptome. The leaves don't change much. Overall, the metabolome doesn't change much under the different conditions. But roots, it changes quite a bit. And interestingly, when plants were grown under heat stress, the metabolome changed the most. So this is 1 day of heat stress. But for 1 week before the heat stress, if they were grown under copper deficiency conditions, it did not change as much. We expected 2 stresses to have a lot more change, but somehow the no copper buffers the change in the metabolome as well as the transcriptome. So why is this happening? And if does this have any phenotypic change? So we did some experiments with Elena later on, and if you are interested, we'll talk about that at the end of the presentation. Now, looking at structural classes, we use this method called Canopus, which instead of predicting from a mass spectrum, this is S-adenosylmethionine, it will say, it will give these classes of metabolites. And then we can say, okay, we can take all the deoxyribonucleosides and see how that entire class is changing under the given condition. The advantage of this method is instead of 5% of the metabolic features that we could identify using traditional approaches, we can go to 40%. In fact, we could go to 80%, and then we removed a lot of predictions after extensive quality analysis. So 40%, it's like a significant bump, right, in how much you can analyze. This is showing the different classes of metabolites, and again, RDPI on the y-axis, showing, and the first 5 are again leaves, next five columns are root. And I'd like you to focus your attention on just these 2 panels. Here, what we found out was flavonoids were highly perturbed in the roots compared to the leaves under all conditions. And then furanoid lignans were highly perturbed under heat stress. So this gives us some hypotheses and models to start digging into the data and associated with RNA-seq, for example. We see flavonoids also, the enzymes also were significantly downregulated in the pathway. Now, ongoing work, we are again, focusing a lot on how do we get better annotation for the metabolome. We don't want to throw away 95% of the data or even 60% of the data. So in collaboration with various researchers across the country, we're trying to develop a ontology system for metabolome, just like gene ontology. So we can do a lot more deeper analysis into suspect. And recently we got funded for International Center for Plant Resilience based out of MSU, but it involves several institutions in the US as well as researchers across the world. The goal of this center is to not only find out new chemicals from plant extracts for agrochemical use, but also find out the genes involved in that pathway, engineer those pathways in yeast and plants, etc., and then test those in field settings. So this is going to be an exciting project going forward. Now I'm going to shift from the metabolome to the genetic aspect of our research and talk about how this diversity evolves, and one at the enzyme level, and second, zooming out again at a very large geographical level. So for looking at enzymes, these are the contributors, these are the main drivers of metabolic diversity, right? We focus on an enzyme family called BHD acetyltransferase. And this enzyme HCT, which is a BHD, does a simple reaction of transferring this fumaryl group to a hydroxyl or an amine group on the acceptor. Now, the reaction is very simple, but BHDs use a variety of different substrates. Some of these are aromatic alcohols, aliphatic alcohols. These are amines, aromatic amines, and more complex structures. And all of these are involved in a variety of different processes in the plant, from reproduction, growth, development, stress response, etc. So understanding how BHDs have come to use these substrate types, that's been a curiosity of mine. That's where we started off this project. And you need to know here that BHDs do not exist across all living organisms. They are, even in algae, they are present in only 1 to 2 copies in the algal genomes, and they have expanded significantly in land plants, dozens and hundreds of copies in each genome. It turns out that in UniProt, there are only 55 BHDs with activities that are deposited. And there are 100 BHDs in tomato alone. So that gives you a scale of how much gap there is between what is annotated, what is there in databases versus where we need to get to. And this is not surprising because we found out a couple of years ago there are only 2 people in UniProt doing plant work. This is not just curators looking at enzyme papers or metabolism papers. Across all plants, only 2 people reading all the thousands of papers published every week and putting them in UniProt. And this is obviously a major constraint for a lot of different downstream activities. We decided to give them a helping hand, at least for BHDs. And Lars, postdoc in the lab, he read 100 odd papers and collected 201 biochemically characterized BARDs, 171 substrates of these BARDs, and put them into 11 different structural classes. Right? So they use a lot of structural classes as a whole. But just to take a step back and think about how much effort this must have taken for Lars, it's a boring job reading papers and putting everything in an Excel file one by one, making sure everything is correct. And so that paper was published in 2022. Nate, who is now a first-year plant biology student, came into the picture last year, and he started doing this job too, and he got bored too. And we had 2 undergrads. We said we can pay you, and they said no, keep your money. We don't want to do this. It was just a boring job. So Nate started thinking about more innovative approaches, and he asked, what if we could use ChatGPT to read papers instead of any humans? Because we can take away their jobs easily. So he came up with this pipeline called Autonate, which enables rapid curation of papers. I actually wanted to call it Autonate, but Nate went the more professional route and called it FunkFetch, which is a much better professional name than Autonate. But essentially, this pipeline reads through a bunch of papers. It screens through thousands of abstracts published in PubMed, and then selects a small set of papers. From each of those papers, it extracts out the enzyme activities, enzyme name, enzyme substrate information, species information, etc. And then we curate it manually. Um, it took a lot of time for Lars to do just the PhDs, but using this pipeline, we illuminated a lot of enzymes using this pipeline. Uh, we were able to screen through 27,000 apps. This is too dark, but the previous lighting was good. We screened through 27,000 abstracts very quickly with a cost of what, $65 actually. And then for 5,500 papers, we extracted a bunch of different attributes like the DOI, journal name, species name, enzyme ID, and bank ID, etc. Very quickly. So this, for all of these 5,500 papers, it costs about $300 or so. Oh, no, actually, a little more. I think 1,200. But from this, we extracted 32,000 rows of activity. Some of these were hallucinations. Actually, this is after filtering for hallucinations. And I can talk about that interesting bit later if you're interested. And these are under active curation right now. Just to give you a sense of how much improvement this makes, we curated this heavily for PhDs, and we submitted the activities to 3 different databases. In UniProt itself, from 55, this goes to 250 pages, a 5-fold jump if we take the curated activities and put it into databases. So, and you can trust what goes into UniProt now because at least 2 and mostly 3 people have looked at each entry, and then UniProt will also look at it. This is going to be high quality. Now that's all technical. How can we use these compiled activities for uncovering new biology? Firstly, we can do evolutionary analysis from these are all the 201 biochemically characterized PhDs that Lars had extracted. And each of the concentric circles here is one of those 11 substrate classes. So we know this clade uses aromatic alcohols, this clade uses anthocyanins and flavonoids and so on. We could define 8 clades based on the bootstrap values, at least 8, but there are many more which are not represented in this figure because they are uncharacterized. Now, we can also go back and see how activities have evolved through their expansion in land plants. So for that, we need to know what is the ancestral activity. And the colors on the branches indicates how far back the orthologous group of that enzyme is conserved. So if it is orange, it means the orthologous group is conserved in angiosperms, gymnosperms, mosses, liverworts, in all of land plants. If it's blue purple, it means it's only present in dicots and angiosperms. Right? So we know these 3 substrate classes, the ability to acylate these 3 substrate classes existed in all of land plants. And this is important because this is involved in lignin biosynthesis mostly. This is involved in waxy ester biosynthesis and polyamine acylation. These 3 features were important for the conquest of land, for terrestrialization, and so BHDs played an important role in that process. We can also then, this particular clade, this is a new activity. It came only in, it existed promiscuously for a long time, for millions of years, but it got fixed in the gene pool only in angiosperms. And we did motif analysis to figure out what is special about these enzymes. This clade turns out that there is a tryptophan, which is unique, and the cysteine, which is unique in this clades. And doing site-directed mutagenesis, we showed that both of these are important. Tryptophan is much more important for activity than the cysteine. We collaborated with Divakar Shukla's lab at Urbana-Champaign, who did molecular dynamic simulations, and basically they showed that the tryptophan, it holds the anthocyanin in place for the catalysis. So the histidine is a catalytic site, the residue, and the tryptophan holds the anthocyanin in place through pi interactions. If the tryptophan is gone, the anthocyanin wobbles around in the active site, and there is no acylation that happens. If the cysteine is gone, The tryptophan also wobbles by itself, so the anthocyanin also wobbles, and there is no activity. So in 100% of the enzymes in that clade, you see tryptophan and cysteine, and so it was important for the evolution of that activity. Now we are focusing on two different plates in these families. One is this very long disordered region that's present only in the lignin acylating enzymes and nowhere else. We've shown that there is some selective pressure uh, maintaining this as flexible. We've changed this to a rigid region by doing side-directed mutagenesis. It increases the specificity of this enzyme towards native substrates. We're digging into that phenotype further. And then going into the broader scale of how do we predict the activities of enzymes, how can we make protein function prediction better? Suping Chen is a first year plant breeding student. She is looking at AI models as well as phylogenetic based prediction of function in enzyme families. And then Xin Yu is a postdoc in the lab who's looking at validating these predictions using traditional approaches like WIGS, enzyme assays, as well as using some synthetic biology techniques. Okay, now I'm going to zoom into zoom out into a broader scale of geographical evolution. For this, again, the model system is different. Here I'm going to talk about Solanum habrocaetis, which is a wild tomato that grows in Peru and Ecuador. Uh, this produces acyl sugars. Acyl sugars are like resin glycosides. They have a sucrose core to which different acyl chains are attached. And they are produced from this one tiny cell on top of the leaf and stem surfaces. And it makes the surface of the leaf very sticky, so insects don't like to lay eggs. Water acts like a water barrier. Um, in different populations of Solanum habrocaetis, the acyl sugar profiles are different. In tomato, there are only 10 or so acyl sugars, but in Solanum habrocaetis, there are a lot more and different between different populations. So Rob Last's lab, back 12 years ago, actually, 2012, they had looked at 55 different accessions of Solanum habrocaetis spread across the range, and they found two major chemotypic clusters for this species. They found out that this enzyme is mutated in some of these accessions, and this acetyl group is missing in these acyl sugar. In one of these, I think supercluster 2, it's missing this acetyl group. Um, when that was mapped out on the species, on the locations of these accessions from where they were collected, all of the inactivations were above this line here. All of these populations are inactivated, but that's not the most interesting part. The interesting part is that the losses occurred by three different means. Here the gene is present, but there is no expression. Here there's a frame shift causes function inactivity. The gene is expressed, but there is frame shift. And here we cannot even detect the asset. So three different means of the same enzyme getting lost. How is that possible, and what's special about this line? When I started working on this project, the idea was very simple. I wanted to figure out where the species originated, how it has moved across the range, and then see how these chemotypes evolved. For this, I did RAD sequencing of 50 Solanum habrocaetis accessions and 4 Solanum penellia accessions. And after I came here, I started analyzing this data along with Jacob Landis, who is in plant biology in the SPECT and Doyle labs. He helped with the population genomic analyses, and even at the genotypic level, we found two different superclusters, again bordering around this line. So that was very surprising, and we spent a lot of time figuring out. I'm ashamed to say that after 5 years of working on this project, suddenly it clicked, and what clicked was looking at the geography of this region. Uh, Turned out that the backdrop, the geography, the ecology had a very significant impact. Uh, need, so this is everything is occurring in the backdrop of this Andes mountain range, which is like a huge wall of rock that emerges and on the South American coast. And there is no east-west movement of species possible because of this huge wall. It's like very tall, right? Um, And there are some parts like this Huancabamba depression, some of these depressions where species can move east-west. Uh, Now, this region that I showed where there are two genotypic clusters, superclusters happening, two chemotypic supercluster happening, that's happening in this region called the Amotape-Huancabamba zone, which is right here. It's one of the depressions. And this region is so, there are so many small habitats, microhabitats, because there are, this region has been influenced by tectonic activity, volcanic activity, Pacific Ocean has come in, gone out, there are riverine beds, weather patterns are different. So there are so many microhabitats that 1/6 of all plant species are found in tropical Andes. Most of those are, many of those are concentrated are due to the endemism present in the AHZ. So this is where all of the chemical and genotypic evolution is happening. The blue dots are southern Habrochaites on the west. So in the southern part of the range, it just is on the western slopes of the Andes. It's a very dry region with hardly any precipitation. And here it sneaked into the Andes and it goes up north to Ecuador, where it is very wet, lot of different coevolutionary processes happening. Where it sneaked in is the southern boundary of the Amotape-Huancabamba zone. That's where it sneaks in, and this is where we see all the inactivations happening. And the Huancabamba depression is where we see the superclusters being separated. The power of this supercluster, the geographical isolation is so high that in the south, we see isolation by distance. That's what you typically see. As the populations move farther out, there is less genetic exchange. So there is there's less gene flow happening because of distance. But in the north, even though populations are very close to each other, like Ithaca and Portland, they are very close to each other, but still there is no genetic flow occurring because of this geographic isolation. This is an example of that. There are two clusters, population clusters. Here, they are very close to each other, but genetically, this is the FST, fixation index tree, which shows how much is the gene flow occurring between different populations. And this is the two clusters here are separated by the Huancabamba depression. There is no gene flow, very little gene flow occurring between these two clusters. And you might see here that I have written SI, here, because when there is geographical isolation, the species is an ancestrally self-incompatible species. But because, I mean, if there is no gene flow occurring and a plant finds itself without any other self-incompatible species members, then it's going to die, that lineage is going to die out. So there is a huge selection pressure for evolution of self-compatibility. So in addition to geography, genotype, chemical evolution, that's another thing that has happened in this species. We worked with Pat Bedinger at Colorado State, who has been working on self-compatibility in the species, and they found that north of the Amotape-Huancabamba zone, SC has emerged from SI 5 times. In the south, it has emerged just once, but two SI-related genes have been mutated several times north of the range, causing 5 independent emergence of self-compatibility, likely because of this geographical isolation of these populations. So we also see all the inactivations of acetylated acyl sugars of ASAT4 happen north of the southern boundary of the ASG. And here you'll just have to trust my word, but this is very complicated. But what we showed here was the 5 different chemotypic superclusters, chemotypic clusters in acyl sugars that we saw correlate very well with which kind of self-compatibility group it belonged to. So it so happened that here we have mixed populations. You can see here MPs, mixed population, which means the self-compatibility alleles exist, but as in heterozygous state. So they have just come about in the geographically isolated populations. And as they went up, the populations got fixed in different corners. And as they went into homozygosity, they may have pulled out the associated ASAT4 alleles along with them, resulting in those chemotypic clusters. So this is very much an example of a founder effect. When you see changes at the genetic or the if you see some transcript is heavily expressed, or you see difference in the metabolome profiles of two individuals, you see some patterns, you might be tempted to think that this is to adaptation. There's some adaptive significance to it. But this is happening just because there's no other way. It's just happening with a genetic drift. One individual got fixed in a population. So this is it should always be thought of as a null hypothesis, genetic drift, and adaptation should be tested against it. And this was a very clear evidence in my mind, at least, although we could not prove any of this, but I think it's happening because of founder effect. In this paper, we talked about the model for Habrochytes population that emerged over here, and then there are two branches that moved north and south, and went through all the chemotypic, genetic, reproductive diversification as it happened. All right. So now I'm going to shift gears and talk about how all of this metabolic diversity and diversification and our understanding of the genes can be applied for improvement of agriculture. We are working on sweet potatoes as one of the major projects in the lab. And you might wonder why sweet potatoes? Because sweet potatoes is not really grown here. Only 200 acres of sweet potato grown in New York. Um, and most of the biggest grower in North Carolina, as well as some southern states. It's a warm weather crop, needs a lot of time for maturation. But there has been a lot of interest in various northern states to grow sweet potatoes, including Canada. Right? So they want to grow sweet potatoes, but this result was very surprising. We grew sweet potatoes for 2 years. In one plot was in Geneva, and one plot was near Albany, and this is showing the yields from the two locations. They were started at the same time from the same stock, and one grew like this, another grew like fingerlings. And this is for another variety. And the second year, it was flipped. It was literally flipped. And I spoke to the extension person who was helping us with this, and they're like, we see that all the time. You grow from one hill to another hill in sweet potato. That's why growers don't like to grow sweet potatoes. They grow it in one corner of the plot because there is so much variation in root phenotypes. But there is an incentive to growing this because of organic cultivation. It can command a premium price. There are no natural pests of sweet potato in New York State, and sweet potato is a huge biomass crop, so we can use it for extracting dyes, colors, health foods, etc. And there is a big industry in the Northeast for health foods. And that was our intent in working with sweet potatoes under colors, how we can use it for these purposes. But one of the main reasons, the main bottlenecks in this process is making how do we make the root growth more resilient to environmental change. And this is a project we started a couple of years ago, so I don't have great results on it, but we have been exploring the use of arbuscular mycorrhizal fungi as a way of increasing the resilience, as well as figuring out the genetic basis of the water stress resistance and resilience and root growth. This is an experiment that Irfan did last year, where he grew sweet potatoes under 3 different water stress conditions, as well as under AMF and no AMF. And of course, the water stress has a lot of effect on growth. That's expected. But even between AMF and no AMF, you can see for this variety, the leaf quality is much better in AMF conditions. In fact, the root weight, we did a lot of phenotyping. The root weight was also significantly better immediately after the stress if they were given AMF condition. So there is some possibility of using AMFs. We also did metabolomic and transcriptomic sampling under all of these conditions, and we are analyzing that right now. Another project on the application side is looking at how we can engineer crops to make novel bioactive compounds. For that, we are using tomato as a model. So tomato fruit develops quite a bit, and there are different varieties which grow to different sizes. Chinu is working on taking the terpenoid flux that goes into making this red color lycopene and diverting it to making drugs which are based on the terpenoid scaffold. So we might have, instead of purple tomatoes, we can have tomatoes making these pharmaceutical compounds. And that's what's going on right now. All right. So this is mostly most of the research that we are doing in the lab. To summarize my talk, I talked about how illuminating the metabolome is important, and the steps we are doing are taking on that front. Discovering enzyme function is one of the major areas in the lab. As I said, we are collaborating with Arjun Kakar's lab at Colorado State to develop novel synbio tools for functional validation. And another area of interest to me is regulation of plant metabolism and how gene expression evolves even how different transcription factors control different metabolic pathways. So that's something we might work on in the future. Engineering plant metabolism, not only for making these useful drugs, but also for stress resilience in sweet potatoes and other crops. And finally, I discovering the genetic networks and biochemical networks involved in stress resilience pathways. Okay, I'll pull out this slide again, and can take any questions. Thank you.
Already described in the paper, or is it also generally new information
Yeah.
for this?
So I can. So the question is whether the FuncFetch tool is extracting information from the papers, or is it also generating new information? So the goal of that pipeline is to just extract information from the papers. Now, Nate discovered some very interesting patterns of how it actually extracts out. So most of the time it extracts out from the paper itself, but sometimes it goes into the references and pulls out activities from the references, which normally we would miss. It's not part of that paper. We also found that it hallucinates quite a bit. Sometimes it pulls out information from its training data. We'll see a paper title saying, it's from barley, oats, and whatever. And the enzyme activity is pulled out is a human enzyme with bilirubin and testosterone as substrates. So it's going into drug bank, which is its training data, and pulling out activities. So some of these, because of the way we are extracting it and organizing it, it's easy to fish out these hallucinations from our data set.
I was wondering for the tomato work in the Andes, where you have the geographical differences, if there was opportunities or some of the collaborators you have left is potentially growing them in different environments and seeing if there are.
Yeah, we wrote a project with Andre Kessler. It was not funded, but essentially that was the goal. We wanted to do reciprocal transplants in Peru and Ecuador and see if that has any adaptive significance, looking at the transcriptome level and transcriptome, metabolome, and the phenotype, actually, if there is any adaptive differences. Yeah, but didn't get funded.
Following up on that, has anyone noted morphological or other differences between those two subpopulations of Capricorn?
Yeah, the there is the leaf differences. There are differences in the growth habits. Yeah.
Thank you, Richard. I just a question about how, what is the time frame that it takes these things to evolve? Do you have any idea? Is it 1,000 years, a million years, or?
I think if Jacob was here, he would have been in a better position to answer this question. We are trying to do some, organize or get some insights on the time scales for these events, but we couldn't do that because of the insufficient data. But I don't know, the two species, Pennelia and Habrochaetes, diverged about 3 million years ago, plus or minus 2 million years from each other. So it happened in the last million or so years. it depends on the selection pressure, right? You could have the traditional Biston Betularia example, the wings of the butterflies changing color because of the industrial revolution and those things. Those happened in a couple of generations. So it depends on the selection pressure and population size. But I don't know in this case how much time it took. Thank you.
The sweet potato, I see, you said that there are no natural pests on the diseases. Is that just because they are in the northern area of the environment or not?
Yeah.
The true varieties of the new?
So there are some wireworm type insects or nematodes that we see when we grow sweet potatoes. We see some holes inside the sweet potatoes, and rodents are the biggest one. They come and eat the farm sweet potatoes quite a bit. But yeah, mostly because it is northern, you don't see the traditional sweet potato wilt or fusarium infections in or the sweet potato weevils are not found here.
This has been a production of Cornell University, on the web at cornell.edu.